A new twin study has added fresh optimism to the idea that nurturing the gut microbiome could play a meaningful role in reducing Alzheimer's risk. While the research is still emerging, the findings suggest that small, achievable lifestyle adjustments, particularly those that influence gut health, may translate into measurable improvements in early cognitive markers, opening an avenue for proactive brain health that complements existing strategies like exercise, mental stimulation, and cardiovascular care. The twin design, in particular, provides a unique lens to examine how microbiome differences can influence dementia risk, controlling for genetics in a way few other studies can. The study followed hundreds of twin pairs over multiple years, focusing on both identical and fraternal twins who were initially cognitively healthy. By examining individuals who share genetic makeup but often differ in diet, lifestyle, and microbiome composition, researchers were able to isolate environmental and microbiome-related factors that might influence the earliest stages of cognitive decline. Fecal samples were collected periodically to map gut microbial diversity and specific bacterial strains. In parallel, participants underwent detailed cognitive testing and neuroimaging to identify subtle changes in brain structure and function. The combination of microbiome analysis and sensitive biomarkers, including amyloid deposition, tau accumulation, and functional connectivity in memory-related regions, offers a rare window into how the gut-brain axis may shape neurodegenerative risk. The most striking outcome was the correlation between microbiome diversity and early cognitive resilience. Twins with richer gut bacterial communities, characterized by higher abundance of short-chain fatty acid, SCFA, producers and anti-inflammatory strains, showed slower accumulation of amyloid plaques and better preservation of memory-related neural circuits. In some cases, a twin with less microbial diversity but similar genetic risk exhibited early signs of cognitive vulnerability, while the genetically identical twin with a more robust microbiome maintained stronger cognitive function. These observations support the growing theory that gut health is not merely a digestive concern but a systemic factor influencing inflammation, neuronal signaling, and metabolic balance, all of which are implicated in Alzheimer's pathogenesis. SCFAs, such as butyrate, propionate, and acetate, appear to play a central role in this process. They modulate neuroinflammation, strengthen the integrity of the blood-brain barrier, and promote neurotransmitter regulation, creating a milieu in which neurons are better protected against stressors that can precipitate cognitive decline. The twin study highlighted that participants with higher SCFA-producing bacteria also displayed lower levels of pro-inflammatory cytokines in blood samples, hinting at a systemic anti-inflammatory effect that likely contributes to the preservation of neural function. This aligns with previous animal and human research, suggesting that microbiome-driven metabolites can influence synaptic plasticity, neurogenesis, and memory consolidation. Diet and lifestyle emerged as practical levers for optimizing the microbiome. Twins who consumed fiber-rich diets, including legumes, whole grains, fruits, and vegetables, consistently exhibited more beneficial microbial profiles. Regular physical activity, adequate sleep, and reduced chronic stress were also associated with a healthier gut ecosystem, reinforcing the concept of multifactorial lifestyle interventions for cognitive resilience. Importantly, these interventions are accessible, low-risk, and empowering. They provide actionable steps that individuals can take today, rather than waiting for pharmaceutical breakthroughs years down the line. For communities, these findings underscore the potential of preventive programs that combine nutrition education, exercise promotion, and stress management to foster brain health from a holistic perspective. The study also sheds light on potential early biomarkers for dementia prevention. Microbiome composition was linked not only to cognitive testing scores, but to imaging indicators of neural integrity, such as hippocampal volume and functional connectivity in memory networks. By identifying these correlations in a genetically controlled twin cohort, researchers have strengthened the argument that microbiome modulation may be a tangible avenue for early intervention. Future strategies could include personalized dietary recommendations, prebiotic and probiotic supplementation, or targeted lifestyle modifications designed to cultivate a microbiome conducive to cognitive resilience. From a public health perspective, the study is especially encouraging because it reframes Alzheimer's prevention in terms of daily choices that are feasible and broadly applicable. Unlike interventions that rely solely on costly pharmaceuticals or specialized medical procedures, microbiome-focused strategies are scalable, culturally adaptable, and compatible with other health-promoting behaviors. Schools, community centers, and senior living facilities could integrate programs emphasizing fiber-rich meals, gut-friendly foods, and stress reduction, potentially reaching wide populations and reinforcing long-term cognitive wellness. The twin design also underscores the power of environmental influence over genetic determinism. Even among individuals with high genetic risk for Alzheimer's, the twin with a healthier gut microbiome demonstrated measurable cognitive advantages, illustrating that genetics are not destiny. This perspective is profoundly hopeful. It validates the impact of everyday habits and emphasizes agency, showing that people can actively shape factors that affect brain health decades before overt symptoms emerge. Moreover, the findings may guide future research into precision interventions. Identifying specific bacterial strains most closely associated with neural protection could inform probiotic development or personalized dietary plans. Coupled with ongoing biomarker tracking, these strategies may one day allow clinicians to recommend tailored microbiome interventions as part of a comprehensive cognitive health regimen. The twin study offers a framework for understanding causality, timing, and mechanism, paving the way for more nuanced, individualized approaches to dementia prevention. In summary, this research paints an encouraging picture. Improving the gut microbiome may be a practical, accessible, and scientifically grounded approach to slowing cognitive aging and reducing Alzheimer's risk. By leveraging dietary choices, physical activity, sleep, and stress management, individuals can nurture a microbial ecosystem that supports neural resilience. The twin study provides compelling evidence that these effects are measurable, biologically plausible, and potentially powerful, even in the context of genetic risk. While more research is needed to confirm long-term outcomes and identify optimal strategies, the findings invite a positive, proactive approach, one where daily lifestyle decisions and mindful attention to gut health can contribute to sharper memory, stronger cognitive function, and a more vibrant life in the years to come.